Well, hello, and welcome to the third webinar in our 2021 Hear from the Experts series. I hope everyone is keeping well as we enter the summer season. We are very pleased today that we can offer you this webinar on generic and biosimilar therapies for MS. We would like to acknowledge PendoPharm for their sponsorship to make this event possible. My name is Beverly McAdam, and I am delighted to host this webinar. I was diagnosed with MS nearly 14 years ago. A classic example of a Canadian woman getting a shocking diagnosis in the prime of her career with two small children who were 1 and 3 years old at the time. I was very fortunate to benefit from the research of the previous decades that A, allowed me to be diagnosed very early in my disease, and B, provided me with medication options. Today, I live with MS, but with effectively no symptoms, although I do continue to take my medication daily. I lend my support to the MS Society, like hosting today's event, to ensure that research continues and the wins in both managing MS today and finding a cure tomorrow are fostered. So before we start, a few housekeeping notes. The webinar is being recorded for future viewing on our YouTube channel. No participant names or identifying information is being displayed. If you have a question, and we hope you do, during the presentation, please type it into the chat box on your screen. We'll have a Q&A session after our speaker presents. We're also streaming live to Facebook, so hello to all of our Facebook Live viewers. Your questions are also welcome. The MS Society does not approve, endorse, or recommend any specific product or therapy, but provides information to assist individuals in making their own decisions. We often hear the words biosimilar or generic in relation to MS drug options. This is a complex and ever-evolving topic. We hope the content today will provide you with an overview of the landscape and help to unravel not only what these and other terms mean, both in definition, but also from a really practical perspective on how it might impact your own treatment choices. So let's get to it. I'm very happy to introduce our speaker today, Dr. Mark Freedman. Dr. Freedman is a professor of neurology at the University of Ottawa, director of the Multiple Sclerosis Research Unit at the Ottawa Hospital, and a senior scientist at the Ottawa Hospital Research Institute. He has published over 250 pieces, including scientific articles, books, book chapters, and abstracts, and has delivered hundreds of lectures and presentations nationally and internationally. His extensive research includes the area of molecular neurochemistry, cellular immunology, neuroimmunology, and clinical studies in MS. Dr. Freedman has over 25 years of experience in the management of patients with MS and has been the principal investigator on numerous clinical trials with new therapeutic agents for MS. So please join me in welcoming Dr. Freedman.
Thank you. I hope you can all hear me. You'll let me know if you don't. This has been a little bit of a challenge today because I am at the hospital and we are open to all the announcements and interruptions that hospitals have, but I will try to get through this all. And if one of those announcements comes through, I just maybe hold my speech so that we're not talking on top of each other. I told the organizers this is not my expertise, and I guess their answer was, well, there isn't a really neurologist that is an expert in understanding pharmacology of generic and biosimilars, but I have been involved as a task force panel to look at these opportunities for several years now, with the same question that came to all of us is, what would it take for someone to walk in with a drug with the same technical name as something we are currently using, and would you be feeling comfortable using it for your patients? And, for drugs that are actual drugs, so like acetylsalicylic acid that we all know has got a brand name such as aspirin, it has a number of other names, but it's technically it's acetylsalicylic acid or ASA, and anybody can manufacture that. The formula is there, and it would be expected that if you could copy the molecule and show simple pharmacology to it that we call pharmacodynamic studies, the half-life in the body, it's eliminated the same way, then the expectations would be that it is the same drug and would work the same way. But when we start talking about biologicals, now we get into a whole different array of molecules, because some of these are actually live in a sense that they have activity when we put it in the bodies. And that's where this notion of a biosimilar came up. Generics are usually referring to the copy of a molecule, such as acetylsalicylic, the ASA analogy I just gave. But when we get into the biological drugs, you could have exactly the same formula, it could even look the same, but it could be biologically inert, meaning it doesn't have any activity once it goes in the body. There's no guarantee. And so for those types of drugs, for instance, one of the first therapies for MS was an interferon. If we simply manufacture interferon, is there a guarantee that if it looks like interferon and flies like interferon and quacks like interferon, to use the old duck analogy, then it should behave as interferon? And that's not necessarily the case, because when you put it into the body, it needs to have that biological activity. And so for many of us, we felt that it wouldn't be enough, and some sort of a clinical trial would still be indicated. Well, why are we even talking about this? And I think you're all aware that the cost of these drugs are extraordinary. And this is a publication just from a few years ago where they looked at the global market for multiple sclerosis drugs, running some 22 billion US dollars a year. And that's just with the drugs that are listed below. We've now got in Canada both daclizumab and ozanimod. Daclizumab was taken off the market very quickly, but there's a drug that's not even on here now, and that's ofatumumab. It just got licensed, and it's similar to ocrelizumab. And these are all very expensive. So the expectations were that this market continues to grow, and you wonder why so many people are competing for this marketplace when you've got a $23 billion marketplace. It's not terribly surprising that there are still lots of interest from pharma to get involved in the marketplace. It's worth so much. Well, my colleague Rory Brown looked at this and published the cost of disease-modifying drugs in a number of conditions. So we looked at things in terms of other autoimmune diseases. You can barely see this on the left, but the point of these curves are they're getting astronomical. They're going up almost logarithmically. And the cost is not coming down, because the cost of generating a new study or a new drug and proving that it is effective in a condition such as multiple sclerosis, the estimated cost is about $1 billion US dollars to complete all studies and bring it to market. And so the graph on the right talks about a number of the different drugs that we're seeing today, including the interferons. So the marketplace is worth probably $1 billion next year. We're starting to see generics come in now. So there's generic competition for fingolimod. We're seeing now new molecules for natalizumab. And this is where the pressures come in. They're expected to push prices up in general. There's probably, when I wrote this slide a couple of years ago, 85 compounds in clinical development. We've got probably over 20 therapies now approved, and there's at least another, I would say, 20 to 30 already in phase 3. Some of the newer agents you know about and have already come out. So siponimod is now licensed. It's no longer in phase 3. Ofatumumab has been licensed. Ozanimod has been licensed. Ponesimod just recently got licensed in Canada. Those studies have been completed. Ublituximab only in the United States. I don't know that they're coming to Canada. And many of these other ones have either, like the OPEXA study was shown up to be negative. The Merck study has just started. Several other trials have just started. The ofatumumab failed, and so there's a long list of meds. But I pulled this off a publication from 2017 you could see right away that that's 4 years ago and highly outdated. One of the reasons the cost remains so high, and this is from an American publication, is that there are profits. Now, thank goodness in Canada, the ability to make money off of therapies for our patients is really not entering into the equation, except in very few circumstances. And to most of us, like myself, we work in academic centers. We have our own infusion centers that are paid for by the public funds. So there's no personal benefit for me to prescribe an infusing agent versus another agent. But in the United States, that's a very different story. In the United States, the profit margin is huge. And I've just sort of highlighted this from this particular publication. The downstream revenue for giving certain medications to patients is highly profitable. And so it's not surprising that infusion-based therapies in the United States are extremely popular, because the physicians or the unit that is prescribing them serve to make quite a bit of money as patients continue to utilize their infusion facility. And this is not the reason for this publication. The reason for the publication was to actually promote that multiple sclerosis clinics are not only good for patients, but they're also profitable for hospitals. This applies, of course, to the United States, not Canada. So there's a whole different regimen. In the United States, hospitals have to show profit. I don't think that's the issue in Canada. I don't think our hospitals make money at all. They certainly go through quite a bit of it. So in terms of the Canadian marketplace for drugs, new brand name pharmaceuticals are expected to increase a lot more than inflation. And the CETA is this Canadian-European sort of comprehensive economic trials agreement where everybody kind of agrees that if you're going to do a study on a drug, that in your country, you have to give it some sort of patent protection. And a lot of this comes in under what's called Bill C-30. But that's one of the cases is to look at what it's actually costing Canadians to be part of this CETA agreement. And this publication from the Canadian Medical Association Journal projected, and this is 3 years ago, to cost Canadians $850 million or more. And it's really to protect the patent. When you think about the development of a drug that's going to cost you $1 billion US, the companies who make these drugs are not going to spend $1 billion if they're not going to make some money. And the only way they're going to make money is to preserve their patents so that people can't copy it and sell a cheaper version. And so they've extended the drug patents to just try to appease the various pharmaceutical companies, to continue to offer the drug to Canadians, but also to do the studies in Canada so that we can benefit early from that sort of ability to perform the research for patients. But this is creating a problem because it, of course, creates a monopoly, and that we're protecting higher brand pharmaceuticals and keeping these cheaper, possibly generic drugs away from the marketplace, and that's not a good thing. So Canada, believe it or not, at least when this study was done, was the third top payer for pharmaceutical products on a per capita basis, with only Japan and the United States being somewhat higher. And at the time with inflation, this number's gone up, and it's about $713 US per person. None of you see this because fortunately we have insurance and we have government plans to put in place to cover the cost of these, but nevertheless, the costs are being paid for this. And when you look at some of the other countries, $515, but look at Denmark, $240 per person. Now, why is Canada paying 3 times as much for drugs than Denmark is? A much smaller nation, albeit. But you can see this on the graph here. Canada's right over here. Here's the United States, and these are all the other countries in the world. A lot of them are paying less for biologics, especially behind the United States. So there is a place for growth here of the non-patent type of drugs. So why aren't these here? Why are we not getting more of these? And I just to look at how the Canadian marketplace works so that you know who's approving these and how the system works. And I do not know the intricacies of this, so this is information I learned in the preparation of this talk. We have various highly regarded health technology assessment agencies, Cadif being one of them. And we, in that sense, we're rated very high, one of the top 15 countries in pharmaceutical expenditure. That plays out in the field because the manufacturers are going to come to Canada and they're going to do the studies here, which benefit, of course, our patients if those drugs ultimately are proven to be effective. But only 22 biosimilar products have been approved since, say, the first agent in 2009 versus the other countries. In Canada, the big HTA is Cadif, and they're based everywhere. They're actually based, I think, in Toronto, but originally they were people from Ottawa. ENES is really Quebec only, but they do exactly the same thing as Cadif. So when a drug comes through and is approved by Health Canada, the first thing that we'll do is go to Cadif or ENES, where they look at some of the pharmacoeconomics and try to negotiate what sort of price would be reasonable to offset the potential benefits, and then make their recommendations for the various provinces to decide if they're going to cover the drug or not. And that takes a long time, usually 6 months to a year before you actually get that. And they make the recommendations, and as you know, wherever you are in Canada, there seems to be different rules, because in Quebec it's one thing, Ontario it's another, Manitoba might be another, and that, a lot of that is based on Cadif. When Health Canada approves a medication, though, the insurance companies are forced to take on exactly how Health Canada says, but they could choose to follow either Cadif or ENES. And so you have now not only 10 different provinces and 2 territories making up their own rules, but you have all the insurance companies doing the same thing. These agencies offer guidance, and they try to get some kind of a cost-effective formula for all of this. I hope I'm not losing people, so before I get into anything more, I felt it's really important to sort of go over some jargon so that everybody knows what we're talking about. And when you read some of this stuff, you'll realize that these are the terms that have been applied. And I'll just run through them. An innovator drug, which rarely you see in the Canadian literature, it just talks about a branded medicinal product, and it may be the original product. So they were the innovator. They're the innovation that has brought the drug to the market. A generic is clearly a copy. It's usually identical in terms of dosage, safety, strength, pharmacodynamics, how it gets in the body, how it gets out of the body. All of that should be identical to the original innovator drug. So it's a generic. But we don't refer to generics if the original product was a biologic, such as the example in MS would be interferon beta in particular. So that's the active ingredient that comes from a biological source. It could be coming from, in the case of one formulation of interferon, comes from manufactured by bacteria. Another one's manufactured by mammalian cells, but it's a live product. So that's the biologic, and its copy is referred to as a biosimilar. I hope I'm not losing you, because when it came to the second drug that we had licensed in Canada, Glutamar Acetate, this drug didn't fall into being it didn't look like a generic, and it didn't look like a biologic, and they didn't know how to classify it. So surprise, surprise, it gets a classification all of its own, and it's called a non-biologic complex drug. And so the referral to it is a subsequent entry, meaning it's come in afterwards, of a non-biologic complex drug. Whoa, okay. But the only drug that has this terminology is Glutamar Acetate. The biosimilars, or subsequent entry biologics, subsequent entry meaning after the innovation, after a patented drug's been released, Biosimilars, and some the Americans refer to this as something else, and Europeans refer to it as something else. So biosimilar, all basically, it's a copy of a biological agent. It looks like the biological medicine, but it's a generic formulation. So that requires of obviously some investment. And as I said, I am a little bit skeptic if someone was just to come to me with saying, "Hey, I just made some interferon and it looks the same, and I, it cost me 1% of the cost of the drug that's currently on the market," and I'm going to go, "Really? Did you test it? Does it have biological activity?" Because I don't know that I believe it, because it's not acetylsalicylic acid. You don't have to just simply make the copy. You need to do something. And so there's some investment in clinical research, but we can't ask generic companies to make the same investment as the original innovator. So they're not going to go out and spend $1 billion running preclinical phase 2, phase 3 trials right up into where it gets licensed, because then the cost is going to be the same. They've got to be able to do less to be able to prove to us at least some minimal level of biological activity without having to invest that $1 billion. And exactly what they have to do is not clear. So they would come to the government and ask, "What do you want to prove to you?" And so in some cases, they would be asked to do some minimal trial to show what's called therapeutic equivalence between the original drug and this new one. And if you do that, maybe that's enough to get it through the marketplace. So there's been some spending to get some of these studies off the ground. And if we look at some of the numbers here in the US, they're quite still astronomical. But when it comes to these non-biological complex drugs, we've had to do something very special. So this really refers to glutamar acetate. And the reason why it gets its own terminology is because, whether you know it or not, glutamar acetate is not really a single product. It's actually a mixture of several different amino acids, all in a different concoction. And in the natural state, these molecules tend to rearrange themselves. So actually, every batch is a little different than the next. And when it first came out, the FDA really didn't want to even approve it because they couldn't show that the same batch was the same. So they had to explain this all to the authorities to show that this is why it's such a unique product. And it kind of, as long as it had about 40% the same, they allowed it to go through. So, and still it's not, you can't make a single copy of it. It's not acetylsalicylic acid. But if somebody just mixes the 4 amino acids together in the same concoction as the original one, should it work the same? That's the question. And so still, a drug like this, even though it's not a simple drug like ASA, and it's not clearly a biologic, it still probably requires something. When we come back to biosimilars, so this is where Cadif comes back again, and they tell you whether or not switching patients would make sense, especially if it's a cheaper product. And of course, patents have been lapsed. So we've given the patent protection for the innovator, and maybe it's 6, 7, 8 years, and now it's past that, and now somebody's coming with a biosimilar. Cadif will evaluate that. They will also look at the price. From Cadif, the next step is to go to PCPA. This is an alliance that works across Canada and advises the various provincial and territorial drug plans. And so they work together with the companies to actually come up not only with a cost, but how they're going to support that. Because some of these biologicals require patient support programs and other things that the original innovator company had provided. And so that has to be given as well. We're not just going to throw the drug out willy-nilly and not give the kind of support that's necessary for all the safety reasons. And then they make their own deals. And that's why it's not surprising to see some provinces preferentially preferring some drugs over the other, especially if you have several different generics. And this is unbelievable. When you go to the US, even with the patented products, they make they cut deals with the various insurers, and say if you prescribed interferon and you were with one particular insurer in the United States, if you wanted, say, brand X, they would say, sorry, we don't have any brand X. We only have brand Y. That's the only one you're going to get through this insurance company, because they cut a deal with the original insurance company, even though we know that the interferons are all slightly different. Well, I mean, here's a quick summary of the various agencies that takes, or really the stakeholders here in driving the costs of generics and the original products. You've got Health Canada, who approve the drug and decide it's effective and safe, and then they don't necessarily put a price on it. The pricing comes actually after the HTA comes in. There's a step in between, right? That's not really in Canada. But the Cadif comes in. INES is the same as Cadif, but in Quebec. And then the reimbursement part comes in, where you get PCPA making some recommendations to the provinces, and eventually comes down to the public and private payers, the private payers being the insurance companies, the public being the exceptional access program, say in Ontario, or the Médicaments d'Exception in Quebec, and I don't know what all the individual names are across the country. But the important thing here is the time. Health Canada can take up to a year to approve a medication, and then typically Cadif will review this for 6 months to a year, and then make a recommendation to the PCPA, and they can spend another 3 to 6 months. So it's not surprising if we hear about a product being licensed or approved in Europe and the United States. Why does it take a year later before we see it in Canada? Here's part of the reason right there. So time to access takes 19 to 23 months to get to a public plan. This is one of the reasons I tell patients, if you're getting involved in clinical studies and it turns out that the drug is effective, by the time you see this drug in the Canadian marketplace, you could have anywhere from a 5 to even, in one case I know, almost a 10-year head start on all the other people who are waiting for this drug to come to marketplace. Hence a good reason to get involved with clinical trials. And this is just showing you the negotiation times it takes across the various provinces to get things together here. And it's different depending on which province you're going to. Obviously, the smallest ones are the ones that have the least amount of time to get through. Let's talk about the non-biological complex therapy such as GA, and this is an example of what has occurred. Now, this is a new drug submission. So Cadif decided that they were going to look at these non-biological complex drugs. So we're referring to the Glutamar Acetate molecule, which has this designation all its own, and said, well, we know you're not ASA, but you're not exactly a biological either, because we didn't get this stuff from a cell line that's made it. So this is why it's an NBCD, but they considered it in the same context as biosimilars, meaning that it's not just enough to show the formula. You need to do something else. You need to show us that there's some sort of biological activities. PCPA won't say anything. They're the ones who negotiate with the provinces. And they just said, well, we'll just deal with it as a biosimilar in terms of the costing for this. So why are we even talking about this? Well, clearly you can't keep going up and up and up like that first graph I showed you, or eventually the costs of just simply these therapies are going to outstrip our public ability to compensate. And the last thing you want is to have a system like they have in some other countries, I won't mention names, where if you have money, you get treatment, and if you don't have money, well, you're out of luck. And hopefully Canada stands for something that's different, and we support people no matter what sort of income they have, and whether or not they have access to private insurance. So what do we have? Worldwide, we have many millions of patients suffering from MS. First-line agents have been around now for 25 years. One of the first-line agents is Glutamar Acetate. And in some other countries that have manufactured very cheap versions, And I've seen this when in my world travels. I've been amazed at what patients are taking. And I said, oh, I didn't even know manufacturer X made that. And they only made it in that particular country. And I said, did they study it? And they went, no, they just made it and they're selling it for, a percentage because in that country they can get away with it. But it's a question of really, is it working? Is it worth it? So these are the annual drug costs for the drugs that we know. Glitect, for example, is the generic form of Copaxone. So you can see already that this is the name brand drug, and this is the cost, the annual cost. And you can see it's not inexpensive, but it's cheaper than the name brand. Okay, and so these are all costs that have come from, I think, the costing a couple of years ago. I don't know if they're really current, and the newer drugs are not even on here. When we look at some of the requirements for a biosimilar, one of the first questions we want to know is truly how comparable are they? And are they just highly similar? And so because you can never make exactly. And most importantly, what does it mean to the patient? So that's the clinical meaningfulness. Is it less safe? Is its impurities causing problems? Because we've seen generic interferons made by certain countries that are containing a ton of impurities, and that leads to a lot of injection site reactions, and people simply won't take them. So what they did is say, well, what's equivalent? How similar do you want to make it? How equivalent is it? Does equivalence have to be 100%? Could it be close? And so you have to actually set limits here. And these are negotiated with the various agencies that do these assessments. And it turns out that the limits are somewhere between 80 to 125%. Now, how can you be 125%? Because of the variability that it goes in many of these types of experiments. That's the whole point of it. And I'm going to show you a study where we actually look at that. And so when we talk about biosimilars being interchangeable, that means you can use whatever the biosimilar is instead of the original product. Sometimes it might even be a better product. There's no reason to think it couldn't produce the same effect and might be even safer than the original one. But it's expected to show the same effect. That's the whole key. Safety and efficacy need to be somewhat interchangeable, and that's where this term has come in. And a lot of it is the results of the studies. If you ask for too much, nobody's going to get into the market and make one because it's going to cost them too much. If you go too relaxed, everybody is going to start making this stuff in their basement and trying to sell it. So you can't be too relaxed on your requirements. And so too stringent is going to be a disincentive to pharmaceutical companies to make it, because if it's going to cost them the same as the original manufacturer, then their expectations are going to be that they're going to make that kind of profit. But of course, then they won't bring the product to the market. So for small molecules, for instance, it's very easy to do this. All you need to do is show pharmaceutical equivalence. This is the example of the ASA. So anybody can make ASA. You can get your own. I'm sure Costco makes their own ASA, okay, and they can sell it as ASA because it's pharmaceutically equivalent. It's going to have exactly the same kind of properties. That's the expectations. But when you have a biological, it's a different story. And bioequivalence is something different now. We have to be able to prove bioequivalence. And proving this is not so simple, especially when we talk about something like the non-biological complex drug as Glutamar Acetate, which is really this mixture of stuff, okay? But this is, for instance, the molecular depiction of a monoclonal antibody, okay? So we have several of these that we're using today for the treatment of multiple sclerosis. Many of you know natalizumab. It's been around for a while, and the most recent being ocrelizumab, and then ofatumumab. Anything ending in MAB is a monoclonal antibody. So look how complex the monoclonal antibody is. Here's acetylsalicylic acid, or aspirin. Look at this little tiny little thing here. Okay, so that you can copy that. That's pretty easy. But how do you copy this? and all the types of things that are involved in this very complex structure involves the way in which proteins are made. I'm not going to get into that here, but these are the kinds of modifications that can all occur when you try to generate a monoclonal antibody. So there's lots of things that could be different about this. Aggregates are sometimes, when you mix this purity, they kind of stick together and then they form aggregates. And the aggregates are really terrible in terms of impurities because they induce all sorts of reactions when you put them into the body. So one needs to look at not only the molecule, but then finally, does it actually work? And so these are very challenging. You can't just depend on animal models. There's no way that we would accept that any more than I have patients who are constantly sending me information about how the animal model for MS has been cured with you name it, and I want some of this. And my answer to them is quite simple. Hey, I can fix that. Come in with furry ears and a tail, and I can give you that stuff because I know it's going to work. But of course, we're humans and we don't behave like the model. So you can't just put it into a model. You've got to go into humans, and as soon as you do that, the cost of even the generic or the biosimilar is going to go way up. There's this very terrible story of actual EPREX. I don't know if you've heard of this story. This is erythropoietin. This is the stuff that they're trying to keep out of the athletes who are going to the Olympics, because everybody wants to take this stuff because it increases your red cell mass, and therefore you can extract oxygen and your muscles will be even stronger. And of course, it's completely outlawed in professional sports. But they made a generic copy of the erythropoietin, and it turned out that the body generated neutralizing antibodies to this new molecule, which then went and negated the body's own ability to stimulate red cells. And before you know it, their entire red cell production facility shut down, and these patients became almost devoid of all red cells and would die. This is what's called red cell aplasia. It's similar to like a leukemia. So you got to be careful when you're making these types of molecules. Demonstrating biosimilar equivalence is not easy. You can't just do pharmacokinetic studies like you can with the aspirin molecule. You've got to be able to show that just getting in and out of the body isn't enough. Did it have the effect that we're looking for? What in MS would we look for? Are you going to do a big study for 2-3 years and look at relapse? Because if you do that, it's going to cost you $1 billion. So what are we going to look for? And so what we agreed on is based on this data, and don't even try to read it, but the whole idea here is that one of the high correlations of relapses is the presence of gadolinium-enhancing lesions on an MRI. So if you could do a study where you take the biosimilar and you put it against, say, the incumbent drug and show that you can get equivalence on the reduction of gadolinium-enhancing lesions, it's inferred that you'd probably have the same effect on relapses. And you wouldn't have to do the 1 or 2 year studies. So this is kind of like the way in which these drug trials are set up. So here's your placebo. This is relapse rate. Here's your new drug. This showed a 70% reduction. That's what we're used to seeing in terms of the superiority. When you look at equivalence, if you did the same experiment and you asked for the innovator, so this is the original product, and here's the biosimilar, how much of a difference would we expect to see in this result to consider it to be equivalent? Well, you have to set limits and you have to decide. And these are negotiated usually with the regulatory agencies. And they would say, here's the 80 to 125%. So here's the one, that's like 100%. So you could be on either side of this and still be considered equivalent. And if you fall in there, you're good. And if your primary outcome is gadolinium enhancing lesions and this is the result, then that would be considered equivalent. But so is that. So you can see how this bar fell on either side, but the confidence limits, this is statistics that applies here, are all equivalent. That would not be. Okay, so that's now, for whatever reason, that confidence limit is too large, and that's probably not something that we would look at. This, of course, would still be considered equivalent. That would not be. So that was now past the lower limit of normal. And so these are all negotiated. So let's go back to non-biological complex drugs, which are glutamar acetate. And there are different products here. Acetates is what was approved in Canada, and it was based on a study. So I'm going to just quickly show you that what was done for this. This is the glutamar acetate. It's a generic. It looks like the same sort of concoction. The pharmacokinetics of this drug are impossible to determine because there's no real half-life of it, because it's a mixture of amino acids that are put together and are in and out of your body very quickly. So the two things that one could look at are some clinical outcomes, but also 100% of people actually develop antibodies to glutamar acetate. So you can actually look at that because that's a biological effect. And so the study was actually called the GATE study, where they tested the generic form of glutamar acetate against the name brand molecule, and they looked at gadolinium-enhancing lesions, and this is the way it was set up, so that there was more patients getting the actual drug. But they did have a placebo arm, which quickly rolled over. The whole study was only really 6 months or to 9 months. This is the original Copaxone arm. This is the Glutect, or the biosimilar, and then a placebo, and then everybody went into this open label extension. It was double blind, so nobody knew what they got, and then everybody knew they were going to go into an open label and then get the drug for 2 years plus afterwards, with the primary outcome being gadolinium-enhancing lesions at month 9. So the reason why it was month 9 is the original Copaxone study did do a sub-study on MRI and did not see a treatment effect at month 6 where all the other products we've used today do. But we started to see it after month 6, and at month 9 you saw clear differences. So this is why the 9 month. Some of you will know, okay, how come everybody else does 6 months? That's the reason. Okay, that was because of the original stuff. Gadolinium-enhancing lesions were the primary endpoint. They looked at other things as well, but they were not powered, meaning they didn't put enough people in to look at relapse rates, because that would have made it a huge study, and that would have cost a lot of money. So everybody agreed that this study would be useful, and this was the EMEA, this is the European authorities. Here are the results. So they looked at now number of gadolinium-enhancing lesions. Those are the lesions in the brain that light up with the dye. And they pooled the Glutect and Copaxone, showing that they were both superior than placebo. You can see how either one of these were better than taking placebo, because placebo would not be expected to reduce gadolinium-enhancing lesions. And then when you look at the equivalence, those numbers that I was showing you, the predetermined equivalence number, in fact, if anything, the ratio was slightly higher. So it was at 1.1. So the actual drug looked, at least in this study, to be somewhat better than the original Copaxone, but it was within the confidence limits that were predefined. So in fact, it was considered equivalent. And then they looked at a number of other things. The immunogenicity is how many people actually made the antibodies to this. And I told you that was one of the biological effects that you can actually measure, aside from just the effect on the enhancing lesions. And you can see the two numbers are almost identical. So the body recognizes it somewhat similarly, and 100% of them make antibodies to it. So I'm going to almost wrap up here. I'm sorry I'm going to leave time for Q&A, and we have about the hour here. And sorry for the technical difficulties we had at the beginning. The importance here is that generics are bringing to MS a cheaper version that might end up saving our society, which is very much a public society for healthcare in Canada. Incredible savings based on continuing to use name brand products. The approval process has to be faster. The development has to be encouraged. We should have some sort of general guidelines that protect us and sort of address general studies that are done with generics, but none of that has actually happened yet. And then we have to also be cautious about the EPREX story, the erythropoietin story, because once this stuff comes out, the name brand pharmaceutical companies, they're always expected to carry out post-marketing pharmacovigilance surveillance. What does that mean? Well, once Health Canada approves the drug, it's all based on the studies, which are, of course, a very select group of patients that qualified for the study, went through, proved that the drug's effective. Now it's released into the community, and doctors are giving it to people not necessarily looking like those that went into the study. So these companies are having to collect data to show, even when it's out in the community, no untoward, unexpected results have occurred. And I'll give you an example of exactly when that happened. When the drug natalizumab came through the clinical trials, everybody thought it was safe. But then we all know that there's this terrible condition called PML, which can kill people as a result of that drug. And that only came out afterwards. So the companies have to look for problems that they didn't anticipate and collect this information and report it to the authorities, for a number of years thereafter. So I think the expectation is when you have a biosimilar, even though it looks like a duck and quacks like a duck, you want to make sure that after you use it for a while, that the ducks aren't falling out of the sky and they're safe. So this is important that they do collect some kind of this information and report back, just like the original companies, just in case there's something that comes up that wasn't expected. So this is a 35,000 foot overview of the generic and biosimilar market with a lot of jargon. I learned a lot from doing the research to present this to you. It is complex, and I hope you appreciate it. But I hope I've addressed a lot of the questions that you've had. It's important that we look for these, and as drugs come to market, we want to try to push for biosimilars and encourage this, because they will eventually lead to the reduction in the prices. And then when the prices come down, everyone can benefit. And I'm not just talking about Canada. There's a lot of places in the world that are very poor, but they have lots of MS patients, and there's no way they can afford brand name products. So biosimilars are going to be effective even in the third world, and that's why they need to be encouraged. It's all about this weighing of costs versus benefit. So with that, I will stop my presentation and maybe stop sharing, and maybe you guys can open the lines, and I'm happy to answer any questions you might have.
Is my sound working? Excellent. Well, I want to start with a huge thank you, Dr. Freedman. You started your presentation by saying you weren't an expert, but as someone with MS, I want to say I truly appreciate your insights and your expertise and the research you did to add to what you would add to the knowledge you clearly already had. So a huge thank you for the time and effort you've put into joining us today. What's interesting is the first two questions are really quite similar, but from two different vantage points. While there were some questions that asked very specific advice and counsel, which we obviously cannot answer on this call, the question is really, and it's one of mine, which is, as a patient with MS, and I am on a medication, I happen to be on Copaxone, what are the questions I should ask my doctor if I get offered or asked to look at a biosimilar or generic? And the second question was actually from clearly someone in the medical arena that asked, what advice should I give my patients? Again, with the same perspective, if a biosimilar or generic is either offered or available, or an insurance company proposes it, what are the questions that a patient should ask? And what are some of the things that a doctor should say?
These are great questions. I hope you can hear me all right. These are great questions, but it's like we don't have control over this. If we had control over it, it would be wonderful. If you write a new script for Copaxone in Ontario, you're going to get Glutect. That's just the way the province decided that they're going to do this. In Manitoba, apparently it's the same thing. Now, if you're on Copaxone and you renew it, that's not a problem. because you've been on it. And right now there isn't a generic form of Copaxone 40, which is the 3 times weekly, although I'm sure that's coming. So in some cases, it's not a choice to the patient. It's really, your insurer may absolutely require it. If it's the provincial payment fund, they may absolutely require it, and you don't have a choice in that matter. One of the problems has become patients who are on, say, Copaxone 40, and now their insurance is gone. Their husband's job is over, they lost their insurance, and now they have to go to the provincial plan. The provincial plan says, oh, you were on Copaxone 40? Sorry, we don't cover that. You're going to have to go back to daily injections. You'd have to make an appeal. Well, daily injections weren't well tolerated. You could make an appeal and eventually get coverage for the Copaxone 40, but not without a fight. So as a patient, it's kind of like you don't really have a choice. Same thing is story now for fingolimod. I believe we are 5 or 6 generic versions of fingolimod. The difference is that's like ASA. All you have to do for fingolimod is copy the molecule, which is actually quite a simple molecule. You don't have to do clinical studies. So that's why we have all these generic fingolimods all over the country right now, which is much, much, much cheaper than the name brand Gilenya, which is now out of patent. I think we're going to be faced with the same thing in a year or two with Aubagio and probably Tecfidera, because these drugs have all been around for a while and copies are being made. So I'd like to think that we have some choice here, but I don't think you do. From a physician standpoint, I want to be assured, as I explained in the presentation, that the proper research was actually done, that the biosimilar is what it says it should be. And if anything has come out subsequent to the studies in these surveillance types of analyses that makes us concerned, that those can be brought to light. Now, so far we have not seen those, but I want to be assured that the companies bringing out the generics or biosimilars are in fact collecting that data and are reporting back to us saying we've seen now 1,000 patients in Canada on this product and no new problems have arisen. Now, from a physician standpoint, it's all about safety.
Thank you so much. So just to recap, if you're already on Copaxone, you may be able to renew the same, but if you're newly diagnosed, depending on the province, you might get something different. A follow-up question that we got on the web was, when you're on a generic or a biosimilar, will you get the same one every time your prescription is refilled? And if you don't, should you ask to have the same one?
We see this all the time with drugs because there are I'm going to say about a half a dozen different generic manufacturers in Canada, and my patient comes back with unrelated disease, say epilepsy. My last drug said Apo something. This one says Teva something. Is it the same thing? It is the same thing. It's just whatever the pharmacist happens to have stocked, they stock the different generics, and you're going to see this depending on who's filling the prescription. And it's up to the individual pharmacist as to perhaps he's not stocking 5 different generics. He'll stock one of them. And if you went to a different pharmacy, you may get a different generic. But theoretically, they're all the same.
That being said, the next question sort of leans into what you mentioned about the appeals process. If you do want to stay on the original medication, can you share any other details that you know about how the appeal process works to stay on a brand name medication?
It too is different in each province. You have to know, and the doctor has to be willing to go to bat for this because we're not paid to do this, and it's extra time in order to fill out all these forms. Sometimes it takes more than one appeal. There's the appealing to the insurance company. There's the appeal to the province, and the forms are different. I find I win more with the province than I do with the insurance companies. The insurance companies just make a decision internally and say, no, we're not funding it, period. And rarely can you appeal that. But the province, you can usually bend an ear. I have been able to get Copaxone 40 for patients who went to that for the simple reason that they were intolerant of the daily injections. Are you on the daily or the Copaxone 40?
Daily.
Can I ask? You're on the daily. So, some people do extremely well on the daily for years and years and years. They don't get any reactions. They don't get the so-called lipoatrophy, little holes and stuff, and they're fine. In fact, they just, make it a routine of every day. Others have had some problems, and they've had to go to the Copaxone 40. I've also had a situation where I've offered the 3 times weekly, and patients said to me, oh boy, 3 times a week, that's like what? Monday, Wednesday, Friday. What happens if I forget? I don't know. It's so routine now that it's like I have it with my orange juice. Okay, I'm going to forget. I'd rather just stay on the daily because it's not a problem for them. For those people, they're going to get the generic. And probably there's going to be a generic 3 times a week, and then we're not going to have this discussion. But I've had to appeal for those people who wanted to stay on that.
Well, I just want to say a huge thank you, Dr. Freedman. As you've gone through the research and how biosimilars and generics are reviewed, you've certainly increased my confidence, should I end up on biosimilar or generic at some time in the future, that I better understand the effort that goes into approving those. And so I thank you for that. And on behalf of everyone that's on the session, it was, you brought some complex scientific concepts to life. And I really, really appreciate that. But unfortunately, that's all the time we have for questions. And I'd like to thank Dr. Freedman. I'd like to thank the folks on Facebook and on the web. We're very lucky, Dr. Freedman, to have you take the time to share your expertise with us. As we wrap up, I would like to highlight some upcoming events, though. There's lots going on this summer. In fact, if you're looking for a summer challenge, you need to get your friends and family on board and set out, of course at a safe distance, on your ride to raise funds for Canadians living with MS. This summer, how far and where you choose to ride is up to you. For more information on MS Bike, visit www.msbike.ca. My personal favorite, at least with my kids, is that we're also looking forward to Burgers to Beat MS. This annual event is happening on Thursday, August 19th, where at any A&W across the country, $2 from every team burger sold will be donated to the MS Society. Last year, Burgers to Beat raised $1.3 million, and since 2009, it's raised more than $15 million. If you're interested in more webinars, our partners at Can Do MS in the US offer monthly webinars, and on July 7th, you can join them to learn about fatigue and other invisible MS symptoms. Go to www.cando-ms.org. Finally, I want to also remind you about the MS Society of Canada's Knowledge Network, which is staffed by trained navigators to answer questions related to MS. You can see on the slide how to connect with them through phone, email, and live chat. You will be receiving a survey following this webinar, and I encourage you to fill it out to let us know if this information was valuable to you. Again, thank you to Pendo Farm for their sponsorship. Thank you to Dr. Freedman and to all of you for participating in today's webinar. Take care and have a great day.